Alright, here today is Dr. Gregory. He's currently a professor of food science and human nutrition at the University of Florida. He received both his bachelor and master of science degrees from Cornell before receiving his PhD at Michigan State. He's been on the teaching and research faculty at the University of Florida since 1977, so a long-time Gator, which pains me as a Seminole. But his research is focused on nutritional properties and metabolic function of B vitamins. And today he'll be speaking to us on probing the effects of human vitamin B6 insufficiency using in vivo kinetics and metabolomics. So please help me welcome Dr. Gregory.
All right, thank you very much, Erin, and thanks to all for being here. Thank you to Susanne for inviting me, and I'm glad we could finally make it after a few cycles of invitation. So anyway, Erin, I took the gator head that I usually had off of my opening slide. So I'd like to talk today about one of the nutrients of long-term interest, vitamin B6. I've been interested in that coincidentally since my PhD project, which my professor said I have this interesting project on vitamin B6 at Michigan State and 100 years ago. So you never know. I've been back to B6 for quite a while now after developing a lot of the techniques that we'll be discussing of kinetics and stable isotopes to probe areas of folate requirements and folate metabolism and bioavailability. They're all working in the same general set of pathways. The other thing that I hope to show you is that as you go along in a career, things that you may be exposed to in graduate school, like amino acids that I never thought I'd be studying, come back to be useful tools. So my advice is learn as much as you possibly can about science, but also about various areas of nutrition, because they're all interrelated in one way or another. So the experiments with humans that I'll be talking about are definitely team efforts, and I've been blessed with some very good graduate students and postdocs in my group that I've named here, and a long-term colleague, physician Peter Stackpool. More recently, some very outstanding biostatisticians. And then as we moved into the area of metabolomics, which is still a developing field, a series of groups involved in that, including mass spec lab at University of Florida, colleagues, and probably the best nutritional biomarkers lab in the world at Bergen, Norway, and colleagues in metabolomics and mathematical modeling at Duke, and some metabolomics colleagues at Emory too. So it's literally been a collaboration that's developed in a real interesting and productive way. So this is my lab group as of last year, and it's a nice outing sailing. That's my boat behind. And they have been extremely productive in conducting human studies, doing a lot of in vivo kinetic analysis and metabolite profile analysis. So I've been pleased with that. So vitamin B6 is one of the water-soluble vitamins. It's a family of related compounds, and as you'll see, what was an interesting chemical project in graduate school has become one of very prominent public health significance. So we have, this pyridine nucleus. substituted in the 4' position. It's either an amine, an alcohol, or an aldehyde, and all of these can either be non-phosphorylated, like pyridoxine is in vitamin pills and food fortification, or phosphorylated, most of them in vivo. And, well, shouldn't have happened. Anyway, the active coenzyme form is pyridoxal phosphate, and as we'll see, that has many functions in amino acid metabolism and a lot of other things. And then we spent quite a lot of time looking at this compound, pyridoxine 5'-beta-D-glucoside, which is a glycosylated form of the vitamin that's very prevalent in plants, and it's incomplete bioavailability, so it needs to be taken into account in assessing that sort of thing, dietary sources. So we won't talk about that further, though, today. So the context of vitamin B6 now is the Institute of Medicine in their 1998 RDA book took the approach of defining adequacy or deficiency based on a concentration of pyridoxal phosphate in plasma. And they say 20 nanomoles per liter or less is considered insufficient or deficient, depending on how they define it. And so that's kind of the working guideline. And I'll tell you right now, measuring nutritional status based on a concentration of some form of a nutrient is a lousy way to do it, because it tells you nothing about functional status. So the field of nutrition in many ways needs better functional biomarkers of status. I would consider, and quite a few others would consider, that to be really an overly conservative mark, and really under 30 nanomoles per liter, you start to see metabolic changes indicating insufficiency. So based on the 20 nanomoles per liter criterion from the N. Haynes study, the most recent one, about 10% of the population would be ranked as deficient. And I don't know what that means in terms of actual risk. However, what we do know from epidemiological data that's been published repeatedly is that people with either low plasma PLP, either under 20 or under 30 nanomolar, or with low B6 intake, those are considered risk factors for cardiovascular disease, thrombosis, and stroke. Under 30 nanomoles per liter, you're more likely to get a stroke than with a higher level of PLP. So it's a real phenomenon, but the mechanism is not understood at all. And this is independent of associations with plasma homocysteine, which is a risk factor, just a marker for potential vascular disease. So low plasma PLP also is pretty well recognized now as being associated with many inflammatory diseases, many inflammatory markers. So things like rheumatoid arthritis, obesity is an inflammatory disorder, and a number of others will be associated with lower plasma PLP on top of effects of insufficient intake or whatever. So there have been several prominent papers on the area of inflammatory relationships with B6, and my dear friend Jakob Sellev from Tufts proposes that there's a redistribution of PLP to inflammatory cells and tissues. Might be the case, we don't know. It's more of a hypothesis. And my friends from Norway, Per Uland, has shown recently increased catabolism of B6, more production of the catabolic product, pyridoxic acid, which I forgot to mention on the structure slide. That could be. And my hypothesis, based on no data, is that B6 deficiency is not an inflammatory condition in its own right, and we've seen that in our own studies. However, the effects of the marginal B6 status are probably aggravated by inflammation, which then can, if there is catabolism or something, further perturb the B6 homeostasis. So this is an area, as you go forward, if you look at the B6 field, be sure to be aware of this inflammation connection. Further context, somebody's counted about 160 enzymatic functions. What is that? Anyway, about 160 enzymatic functions of PLP. That may not be 160 enzymes, but different reactions involving PLP as a coenzyme. Most of the amino acid metabolism, catabolism, interconversion reactions. In the course of doing those, there are organic acids produced, which can feed the TCA cycle, things like that. Tryptophan catabolism, we'll see later, a whole bunch of other things. And so the interesting thing in assessing effects of B6 deficiency that I am interested in and needs to be developed further is what's the hierarchy, what goes wrong, what's the sequence of events at certain levels of B6 status. So that's a high priority in figuring out disease nutrition correlates with respect to B6. So one carbon metabolism, in the area of metabolism, the interaction of three vitamins, folate, B6, and B12, is critically important in a series of cycles that involve either the generation of single carbon units for nucleotide synthesis, and that is purines and thymidylate, needed for DNA and all sorts of other things, and single carbon units for cellular methylation processes. So the way this works is various donor molecules, such as the amino acid serine, glycine, a few others, can enzymatically donate a carbon, like the serine side chain, to a folate. This is tetrahydrofolic acid. To the folate then acquires this carbon unit. So this is methylene tetrahydrofolic acid. This PLP is pyridoxal phosphate. It's a B6 catalyzed process. And then a reduction, methylene tetrahydrofolate reductase to 5-methyl THF, which is its only function is a B12 dependent reaction donating the methyl group to the amino acid homocysteine. So it's a way of recycling methionine from homocysteine. It's also a way of regulating the concentration of homocysteine. So in B12 deficiency, that would be impaired, for example. So this reaction is vitally important. It requires adequate supply of THF, folate, but it also potentially is dependent on B6 for the supply of the one carbon units. Okay, glycine also is important. There's a mitochondrial glycine cleavage process which breaks up the glycine molecule to generate methylene THF. Okay, in the methylation process, methionine becomes S-adenosine methionine. There are 100 or so methylation reactions, including DNA methylation, creatine synthesis, phosphatidylcholine synthesis, a whole bunch of other things. After that methylation process, transmethylation, S-adenylyl homocysteine generates homocysteine again, and some of it goes through another cycle of remethylation. Some of it is catabolized down this process called transsulfuration, which involves 2 PLP-dependent steps. So you see there's really integration between these 3 vitamins, 4 actually if you consider the flavin in the MTHFR reaction. So in B6 deficiency, the sequence of events here was not really very well understood. In folate deficiency, we spent a lot of time doing modeling and in vivo kinetics in people to characterize those, and that's a different talk. So we'll move on with B6. And so that's keep that in mind. We'll come back to it. Folic acid, if you eat, fortified grain products, that feeds into DHF and then THF. So that's the function there. It's a direct supply of folate. All right, so the last slide was an oversimplification. There's actually mitochondrial one carbon metabolism, whose job it is to generate formate, this little single carboxylic acid, that diffuses into the cytosol and becomes a one carbon source in its own right. And we showed that that's actually a very important reaction quantitatively. And then the latest event in one carbon metabolism is this. Patrick Stover at Cornell showed there's actually nuclear one carbon metabolism to generate thymidylate as needed in S phase of cells. So, what seems like a well-known vitamin, it just keeps getting more and more complex, and the interactions with other vitamins equally complex. Okay, so in 15 years ago, 1998, 99 era, we came up with the hypothesis that marginal status for folate at that time and then marginal status for B6 would perturb the various metabolic events in one carbon metabolism by impairing essential or insufficient essential coenzymes and or folate substrates, thus reducing the flux of essential reactions. And flux, that means just the in vivo rate of a reaction or of a pathway. And the result of the reduction in flux is that there could be then insufficiency of essential metabolites, like nucleotides, like various other products, methyl groups. So that was the hypothesis: reduced flux of reactions as a result of vitamin insufficiencies and concurrently changes in patterns of metabolites. Turned out that was wrong. So experimental approaches we've used over the years, we've done a lot of controlled dietary studies. So they have their limitations, but they also have their strength in that you can do absolute nutritional control and minimize the variability among people and also know exactly what they're eating. And you can do before and after nutritional studies, which most of ours are. The then stable isotopic studies can be very focused and targeted at certain pathways, and although they're small studies, they can be really informative. limitation is that, say, we're feeding somebody a low folate diet or a low B6 diet for a reasonable period of 4 weeks or 6 weeks or whatever, that doesn't reach steady state equivalent to a lifelong bad diet. But it's the best we can do, and we infer a lot from it. We've also done some observational studies of populations looking at status related to metabolite profiles. I'll show you one of those. Mathematical modeling, we've done some of that with colleagues at Duke, and I'll not talk about that much today. Okay, so questions in our restriction studies. Does B6 restriction change the flux of PLP-dependent processes, and what metabolite profiles change, and are any of those useful biomarkers? Can we infer anything about what's going wrong? And what can we infer about metabolic control? So look at that. First of all, I want to mention something about stable isotopes. Stable isotopes are heavy forms of atoms. There are many forms of many elements who have a mean form and then a non-radioactive isotope such as hydrogen with mass of 2, it's deuterium, there's an extra neutron, it's not radioactive, and in nature it's just a tiny bit of natural deuterium. For carbon-12 is the main carbon isotope. Carbon-13 then is the stable isotope. About 1% of all the carbon in the universe is carbon-13. We can make, as you will see, or now buy, compounds that are synthesized with certain positions enriched in carbon-13, so that they have the same metabolic properties as the unlabeled compound, but have a different mass. So if it has 3 carbon-13s incorporated into the compound, it would have a mass of 3 greater mass units that can be detected by mass spectrometry. Okay, so principles. So we work with human subjects. That's my one gator thing there. So in normal metabolism, substrates going in tracer experiments, the tracer people use lingo like trace E, the compound that's being modeled. So if a certain amount is being incorporated into the body by diet and or synthesis, then that same amount would be coming out by metabolism or excretion. So let's say 20 micromoles per kilogram per hour in, same would be coming out. So that's a steady state. So if we do an experiment with a stable isotope labeled compound like serine, the amino acid might have carbon-13 incorporated in it. That would be in a sterile pyrogen-free IV solution. So in our clinical research study, that would go in one arm in an indwelling catheter, and there would be another in the other arm that can take timed blood samples. And if we're so inclined, they can take breath samples, just breathe into a tube and determine the isotopic enrichment of breath. So if we're doing this in fusion, the same non-labeled metabolism's going on unperturbed, but we're putting in a small amount of stable isotope, the green dots, that mixes in vivo in the tissues and cells. And if there's metabolism, then the proportion will be the same mixture as here in the body. And if we then take blood samples, we can measure by, in our case, gas chromatography, mass spectrometry, the enrichment, which is simply a molar ratio of the labeled compound or its metabolite to the unlabeled plus labeled compound. So this would be just in a percentage or mole percent excess above the natural abundance. Okay, so we can then, if this is 20%, what we're trying to figure out is what the unlabeled metabolism is, and we know what's going in, we can infer then what's going on with the unlabeled. So if we're doing an infusion, these are called primed steady state infusions or primed continuous infusion protocols. We get a priming dose, just an initial bolus, followed by a continuous infusion. And so enrichment of what we're infusing, the tracer, will go up rapidly and then plateau. So we want to know what this plateau is, because we can use it to calculate the flux of the compound. So we're infusing this serine, for example. We want to know serine flux, then we simply calculate the infusion rate going in, and you can either divide just by the plateau enrichment, which might be 10% or less, or a little more elaborate equation that gives the same number to calculate the flux. So we can say serine flux is so many hundred micromoles per kilogram per hour in the body. Well, that's not real informative, but we'll see how that would be used. And then a product produced from the tracer would show a curve like this, and we're interested in determining the slope, the initial rate here, and then can use that to calculate a fractional synthesis rate. So that's the fraction of the total pool formed per unit of time by metabolism. So it might be something like, for homocysteine, 25% of the pool per hour, something like that, from the precursor. We can then do one more thing with that number, and then that's multiply to calculate an absolute synthesis rate. So the absolute synthesis rate is the fractional synthesis rate multiplied by the concentration in the cell or in the blood, and that's how much is being produced per unit of time. So such kinetic experiments like we just showed are really important, and more important in many ways than single measurements. They can tell you what the in vivo rates are of metabolic reactions. So that's the importance of kinetics. And they can tell you about metabolic physiological processes that can't be observable by static measurements. So we can measure somebody's homocysteine and find it's elevated. Why is it elevated? Can't tell from just a concentration. What we could tell from our kinetic experiments, though, when we did folate studies, was it's not elevated because of any alteration in the folate cycle called the remethylation process. It's elevated because there's more production of homocysteine, which is entirely unexpected. You couldn't tell that any other way. And the other beauty of stable isotopes is that they can allow you to do multiple experiments simultaneously. So we'll be, in the next couple of slides, be looking at an experiment where we infuse simultaneously serine, methionine, and leucine, and none of them interfere with each other if they're labeled correct positions. And then you can measure processes going on in vivo involving all these three tracers. And just like buying a boat, every protocol is a compromise. Some boats go faster, some boats are cheaper, some, all the different compromises, exactly the same with these. You hope you pick the best hypothesis, the best compromise, though. Okay, so let's talk about B6 and one carbon metabolism, and how's again, back to our questions, does it affect metabolic rates or fluxes? Does it affect concentrations? Typical experiment would be healthy young adults. We would give them a 2-day stabilization diet after verifying that they were in fact healthy and passed all the clinical screen, which was just to stabilize protein intake, stabilize vitamin intake, and so on. And then we would do while they were in adequate status, the infusion study and take blood samples for metabolite analysis, and then feed them a controlled diet. In our B6 experiments, we used a diet developed by another group 20 years ago, which feeds a controlled diet with 0.35 milligrams of B6 per day. And that's about 1/4 of the RDA. People would die if they ate that forever, but this is a really good, effective tool to, in a 4-week time, get people to around the 20 nanomolar cutoff, because we want to see what's happening at that level. And then after 4 weeks of that, we repeat the blood sampling and the tracer infusion and timed blood samples from that. And then give them a bottle of vitamin pills and pay them, and they're on their way. Okay, and just to remind me, we've also done just the opposite approach in our most recent studies, where we take people that we were looking at oral contraceptive users that some subset of which were naturally low in PLP, do the study to assess what's going on with them, then supplement them for a 4-week period, and then repeat the blood sampling and the infusion. Okay, just to show that this protocol works, these are our starting people. This is 23 people from a couple of different studies. Plasma PLP in a perfectly adequate range, most of them, and one of them was not, although they were when they were screened. And so anyway, they all go down as a result of this diet, and the mean was right where we wanted it, and some were in the marginal range, some were in the deficient range, and it allowed us to assess the significance of this insufficient status. Okay, what do we know about B6 dependent enzymes in terms of activities? Well, we've done a lot of rat studies, and somewhat hard to interpret, correlate rats to humans, although we know that the PLP dependent enzyme, serine hydroxymethyltransferase, that takes that serine carbon, puts it onto THF, is markedly reduced, both mitochondrial and cytosolic versions, and in lymphocytes, in human lymphocytes after the B6 restriction, it's down about 40%. So it's a good way of evaluating the significance of this reduction. The transsulfuration enzymes, the second one, cystathionine gamma lyase, is reduced in a big way. Mitochondrial glycine cleavage, we predict with modeling that it is, although it's hard to study directly. And so, and most of the other enzymes in B6 in amino acid metabolism are as well. So the tracer, methionine tracer, fully labeled with carbon-13. It's denylated. It goes through the methyl donation and becomes adenylyl homocysteine. Homocysteine dissociates from that. The initial methionine was fully labeled with 5 carbons. The homocysteine has 4, and then if we have an unlabeled methyl group and 5-methyl THF, this B12 dependent remethylation process, then we'll put the unlabeled methyl group here, so the methionine becomes now with 4 carbon 13s. So in our mass spectrometry, we look at the different masses of methionine. Unlabeled methionine is the M+0 methionine that's gone through the cycle, and then remethylation is the M+4, and the infused methionine M+5. You can tell all that from the mass spectrometry. Same with the serine. Serine is labeled on the side chain. If it goes through the SHMT reaction, the product is glycine, which is no longer labeled, but the folate is carbon 13 labeled, becomes 5-methyl THF, carbon 13 labeled, and then if it goes through a remethylation of unlabeled homocysteine, that methionine becomes singly labeled. So the appearance of singly labeled methionine in this experiment tells you that it came from serine. So we'll be talking about that. So the bottom line is you can do these experiments simultaneously with the methionine and serine infusion, which then, again, in the mass spectrometry, if you see the M+1 methionine, that came from a remethylation from serine, which is the PLP-dependent reaction, or depends on it. Okay, typical data, you get tons of data from these experiments. Each one of these is a timed blood sample, and these are enrichments or fraction of labeling determined by the GCMS. Here are our infused tracers in the infusion solution. So the leucine, which we use basically just as a monitor of muscle turnover or protein turnover, and the infused serine and the infused methionine. Metabolic products from that, from which we can calculate things like the fractional synthesis rate, is here's the methionine produced from the remethylation from serine, homocysteine from the infused methionine, and methionine M+4, that is the product from the methionine cycle. So to make a very long story short, from a long series of these experiments and infusions, including the 3 tracers I mentioned, and also a series of glycine experiments with the same model, we can conclude a few things. One is that the B6 restriction at most causes a modest reduction in the glycine cleavage flux, which is a B6 dependent process. The methionine cycle, that cycle of remethylation, transmethylation, was pretty much unaffected by B6 restriction. All this is contrary to my assumptions. Transmethylation, transsulfuration flux, remethylation flux, and transmethylation flux all were unchanged. And bottom line is pretty good evidence that the initial hypothesis was wrong. B6 deficiency, even though there are 4 or 5 essential PLP-dependent enzymes in one carbon metabolism or reactions close to it, substantial B6 restriction had no effect on fluxes. So this, I think, ties into many of the aspects of the pretty dynamic field of metabolic control theory of what changes in certain enzyme activities and what happens to overall pathway fluxes might be entirely different. So these, what I thought would be sufficient changes to induce big functional effects, were not showing up in terms of measured in vivo kinetics. So what did change? Well, a lot. First of all, if you look at some individual variability, we've been talking about group means so far. I don't know if there will ever be personalized nutrition, but you can sure see personalized kinetics from these kind of studies. First of all, I haven't mentioned yet glutathione. The PLP-dependent transsulfuration pathway goes to here, makes cysteine. Cysteine is one of the 3 amino acids for glutathione, the cellular antioxidant. And so, in the course of doing a glycine infusion experiment, my smart friend Dwight Adams at University of New Hampshire, or University of Vermont, said that they could measure the isotopic enrichment of glutathione from our glycine infusion experiment. So we measured glycine enrichment in red blood cells. We measured kinetics in other things and sent red blood cells to Vermont for the measurement of the enrichment. And what you see is that glycine does in fact enrich glutathione in a measurable way. This is in red cells because it's easy to look at glutathione synthesis. So bottom line, if you then look at group means, there's a small effect of B6 restriction on the group mean of fractional synthesis rate and absolute synthesis rate. But if you look at the individuals, there's a subset of, well, about half of them either stay the same or increase in their rate of glutathione synthesis, and approaching a half decreased a lot. So I don't know the implications of this, but if you look here, this is absolute synthesis. I would rather be one of the persons that didn't have their glutathione changing or made it increase than to be one of these people where it cut in half. And so I think their ability to, fight oxidative stress or do drug detoxifications, this is also involved in, could be impaired in B6 deficiency. So another example, this is cystathionine production. One out of this group of 9 people dropped a lot. We don't know why this kind of variability exists. And to point out, looking again at the SHMT reaction that requires this, whoops, sorry, that depends on PLP for the acquisition of serine to then remethylate homocysteine to make methionine. This is remethylation flux through this entire pathway. If B6 was limiting the pathway, you would see then a lower flux. And what we see is most don't change or even increase in B6 deficiency, which is contrary to what we know about metabolic control, but also a few declined substantially. So the means don't really tell the story at all. Okay, I'm going to move along here. So we were also interested then, after finding that in vivo kinetics really were not changing significantly, what was changing, we had opportunities in the growing field of metabolomics. Global metabolomics is applying a technique like NMR or mass spectrometry to measure as many compounds as possible and then sort out and make inferences. Targeted metabolomics or metabolite profiling is measuring certain concentrations. So bottom line, this is an example of an NMR spectrum of before and after B6 restriction, which is very difficult to interpret, but a number of changes were occurring, which were, if you look at the entire NMR spectrum in a multivariate analysis where the entire spectrum of one individual is represented by one symbol, this is restricted and adequate. You see there's really statistical evidence the entire spectrum was different. And what was different about it, some individual compounds, most of which were either organic acids or amino acids, and a few oddball things that we can't figure out that are not B6 dependent, like proline and TMAO. The targeted metabolomic profiling or metabolic profiling, I think, is more informative. We have used techniques to measure one carbon metabolites and also tryptophan metabolites, which have been known in the B6 area ever since the 1930s when pyridoxine was first discovered, and tryptophan catabolic products led to its discovery. So all the yellow boxes can be measured by an LCMS technique, and so it fills in a lot of the places in one carbon metabolism. And then if we measured the folate compounds by a sensitive HPLC procedure that we have, we can account for much of the pathway. So in doing that, what do we see? Well, two important things are changing, actually several. Serine and glycine dependent on the SHMT reaction, and cystathionine in marginal B6 deficiency. Cystathionine in your blood will double. And so that's a very sensitive biomarker. Glycine and serine less so. The tryptophan catabolic pathway, tryptophan forms kynurenine, and then kynurenine goes through a reaction catalyzed by this enzyme that's very hard to say, kynureninase, that's a PLP-dependent reaction, or the kynurenine aminotransferase. All these things can then go through further PLP-dependent reactions, and so you get a pattern of metabolites of kynurenine and related compounds. And this goes all the way down to NAD and nicotinamide and so on. So my Norwegian colleagues measured these in our B6 restrictions, and we see large changes in kynurenic acid, 3-hydroxykynurenine, which turns out to be a very good biomarker. And the nicotinic acid was later found to be spurious, and they're certainly not niacin deficient. So anyway, perturbations in the tryptophan catabolites. So if you throw all the data together of one carbon metabolism and tryptophan metabolism into a multivariate analysis, what we see is that there's very large separation of groups indicating large differences in the metabolic profile. And the orange symbols are the ones that reach statistical significance, but the overall pattern was significantly different. So, can we use this clinically, or can we make a diagnostic tool that assesses functional status from a handful of these compounds? And I think absolutely, that's a high priority in my work for the next year or so. So, just as an example, we were screening a bunch of women for our oral contraceptive study. So we did the full metabolite profile, 1-carbon and tryptophan metabolites. So the orange dots are the highest ones. Those are very likely supplement users. This is perfectly adequate B6 status. The blue ones and the gray ones are the low ones. What we see is clear separation of groups, indicating there's something about their pattern of metabolites that's different, and also that of the gray ones, the low PLP under 30 nanomoles per liter, some of those are no different in their metabolic profile than the adequate B6 deficient, but some of those are. What that says is that low PLP under 30 nanomoles per liter is a lousy predictor of metabolic conditions and status, but that the metabolic profile probably is the pattern of metabolites. So this is just the lows versus the highs in PLP, and they're clearly different in their pattern of these metabolites. What's different? I think we could certainly take the 3-hydroxyquinurnine, cystathionine, and a couple other things and come up with very sensitive biomarkers. Each one's pretty good on its own, but the whole thing, I think, would be a very useful diagnostic tool. Let's skip that. So, in conclusion, even mild deficiency of B6 causes many metabolic changes in these PLP-dependent pathways, and some effects that are secondary to them. There's surprisingly small changes in pathway fluxes as far as population, group means from our studies, but certainly some individual effects. And I think the targeted metabolite profiling certainly complements global metabolomics, and it also complements kinetic measurements of flux and figuring out if the flux is not changing, what is different, and the reason that it's different is because the metabolites are compensating, and I can explain why that is on a kinetic basis later. We're out of time. But the flux is being driven by alterations in metabolite pools. So, unlikely diagnostic importance of these biomarkers, I think that's one of the practical outcomes of this. So, the 19th century painter Winslow Homer went to Florida and fell in love with sailing, and I did too, so I like to end with this slide. So that's all. Thanks very much for your attention.
We'll open up the floor to questions.
Yes.
In the study where was most presented with the glutathione synthesis that decreased with the marginal PLP deficiency, but only half the population, you're saying only half the population experienced
Yes.
decrease. Did you have the opportunity to look further into how compliant those participants were with the dietary intake, what the severity of their deficiency was?
Yes.
And if so, what did you find?
Yeah, that's a good question. Humans are inherently non-compliant, so you have to do two things. One is educate them about the importance of complying with the experimental diet. Second of all is convince them that you can find out if they're non-compliant, and we can by, in a restriction study, PLP should go down steadily, go down fast, and then continue. If it doesn't, you know that they've been non-compliant. Same with folate experiments and things like that. And the last is convince them of the importance of their participation in the overall conclusion and something that has public health significance. And we have had very, very few of, a lot of human subjects over the last 12 years or so, very few non-compliant people. So we're very good. You just have to educate them, work with them, and have a tool for assessment.
So just to follow up with that, were the people that didn't decline as much as the others, that half the population, when they started, or their level of PLP insufficiency, where was it on the scale between 20 and 30? Were they higher? Were they closer to 30, or
the ones that didn't decline as much,
or was there no difference?
they all seem to decline to about the same level. So they start off at a pretty wide range of adequacy, most of them. They come down to a pretty narrow range. So as far as percentage change, I don't recall that, but the diet's a really good tool for just getting them in the range of low PLP that we want. So I think that's just the best tool we have. What the limitation of this, I mentioned there are limitations in every study, is that this is not the same as a lifelong bad diet that doesn't have any, poor sources of B6. It also is not in a population that has chronic inflammation that many, people that have B6 insufficiency do. So, good questions. Thank you. Yes.
In your research, because most of what I ever hear about is B6 overuse. Have you seen anything in your research with insufficiency and neurological function?
Not much in insufficiency. What I don't recall is that in certain neurotransmitters, in tryptophan metabolism, the side that goes into neurotransmitters, there's another B6 dependent step there, the decarboxylase. And what I don't remember is the association with neurotransmitter levels or function and B6 status, although somebody has studied that. If that's another good question, and I'm sorry, I don't know that one.
Thank you.
Yeah.
Is that like you tell them not to eat something, or you tell them exactly what to eat?
No, they're fed a diet at our clinical research center prepared by our research dietitian. My former postdoc described it as a junk food diet, but it's a carefully controlled junk food diet. But it's adequate. It's designed to be adequate in protein and essential nutrients. We give them a supplement that's custom made that contains everything except pyridoxine. So we hope they're in good shape. Yes.
So first you showed flux data on a certain part of the metabolism. Then you went on to concentrations, but now I'm thinking you show different types of changes in the concentration of certain substrate, which you did not measure the production with the stabilizer tool.
That's true.
I'm wondering, what is then your next step? Going back to the flux measurement onto tryptophan conversion?
That would be a useful thing to do. My last venture to NIH, my work in the area of B6 was rated as low, whatever their word is, not impact, but something. So they didn't think there was enough B6 deficiency in the population to merit further study. I disagree with that heartily. But, I may or may not be able to do that kind of study depending on if there's funding for it. But, we have a good mathematical model of tryptophan metabolism. So we haven't done the actual flux measurement. So.
In those people who do not eat animal products, like vegans,
Are you aware of any studies on glutathione synthesis in red blood cells or in the whole body?
I'm not. I don't know if anybody's done that.
The reason I ask this question is in the past 2 or 3 weeks, the National Dietary Guideline Committee recommended Americans will not eat lean animal products in the diet. I don't know if these people would know the consequence of this recommendation.
Yeah. Probably lower protein, lower available B6 for one. I don't know. That's a good question. But the first question, no, I'm not well aware of any other glutathiones.
One last
Yes,
question.
okay. John, you have.
Well, that must have been a good question because I was going to ask you about liver and muscle levels of glutathione given.
I don't know those at all.
Still don't know.
So, yeah.
But with vegans, et cetera, that would be a great population.
Yes.
Could be better than
Well, I guess we'll conclude now. Let's have a big hand for Dr. Gregory. The student luncheon is back in Cater Matil, the main conference room, and I just forgot the